السلام عليكم طلبة الصف السادس الإعدادي في مادة البيولوجي لطلبة المدارس المتميزين وكلية بغداد. نبتدئ مادة البيولوجي بالفصل الأول chapter 1 لموضوع الخلية the cell. عندنا chapter 1 the cell الخلية. نبتدي بموضوع introduction. What is a cell? The cell is considered as the basic unit of all organisms. Generally, there are two types of cells from a structural point of view. إذن نعلم أن الخلية هي الوحدة الأساسية التركيبية والوظيفية لجميع الكائنات الحية. So the cell is considered the basic unit. What is a cell considered? It is considered the basic unit of all organisms. So when we want to define a cell, we say the cell is considered as the basic unit of all organisms. Generally, there are two types of cells from a structural point of view. They are the prokaryotic cells as bacterial cell and the eukaryotic as plant, animal, fungi, and protists. So the cell is the basic unit. We know two types of cells in general. The first one is the prokaryotic cell and the other is the eukaryotic cell. What are these cells and what's the difference between the two of them? إذن ما هي هذه الخلايا اللي هي الخلايا بدائية النواة والخلايا حقيقية النواة اللي هي قسمت إليها أنواع الخلايا بشكل عام. الاختلاف الرئيسي هو الاختلاف التركيبي فيما بينهما. إذا نجد أن الخلايا البدائية والخلايا الحقيقية. كلمة Prokaryotic برو وكاريو. برو هي ما يعتبر بالقديم أو السابق أو ما يسمى بالبدائي، والكاريو هي النواة. إذا نواتها بدائية. يوكاريوتيك، يو بمعنى حقيقي أو حقيقية، وكاريو أيضا هي نواة، إذا نواتها حقيقية. فما هو النواة البدائية وما هي النواة الحقيقية؟ Usually at breakfast, people use an egg for breakfast, a chicken egg. When we break open the egg in a pan, we will realize that an egg looks like that. This egg is a true cell, and it is a single cell. It is a type of eukaryotic cell. Why? Because when the egg looks like that, it has this part, which is the yellow part, and this is the white part. The yellow part usually we say that is the nucleus and not. And the white part represents the cytoplasm. This egg is surrounded by a plasma membrane. إذن الخلية الحقيقية مثال عليها لدينا البيضة. عندما يتم فتح هذه البيضة نجد أنها مؤلفة من جزء واضح لونه أصفر يسمى النواة، أو عادة نسميه نحن المح، ومحاطة بمادة بيضاء التي هي السيتوبلازم تعرف بالأح. ومحاطة هذا السيتوبلازم محاط بالغشاء البلازمي، بلازما ممبرين. إذن بيضة الدجاجة هي مثال على الخلية الواحدة التي هي خلية كبيرة الحجم وهي حقيقية النواة. لماذا نقول عنها إنها حقيقية النواة؟ لأن نواتها هنا محاطة بغشاء نووي. So the nucleus is surrounded by a nuclear membrane. Because the nucleus is surrounded by the nuclear membrane, so it is considered a cell that has a true nucleus, eukaryotic cell. Sometimes when we crack open the egg, we realize that this is the cytoplasm that is surrounded by the plasma membrane, but the yellow part, as we said, the nucleus, is dissolved in the white part, the cytoplasm. When that occurs to the chicken egg, that means the nuclear membrane has been ruptured. So this nucleus is not surrounded by a nuclear membrane. When that happens, we cannot return this nuclear matter back to be in the form of the spherical form. Here the nucleus shows the spherical form, while here there is no nuclear membrane. That means it has been ruptured. This is similar to the prokaryotic cells like bacteria, because bacteria has a plasma membrane. Also, it has cytoplasm, but the nuclear matter is not surrounded by a nuclear membrane. So because this nucleus is not surrounded by a nuclear membrane, so this cell is known as a prokaryotic cell. That means it has a primitive type of nucleus. نوع النواة نواتها بدائية لأنها ليست محاطة بغشاء نووي. إذن بالنسبة للبكتيريا which represents the prokaryotic cells نجد السيتوبلازم والغشاء البلازمي والمادة النووية ليست محاطة بالغشاء النووي، نسميها نيوكليويد، لأن المادة النووية ليست محاطة بالغشاء. إذن هذه تمثل الخلية البدائية وهذه تمثل الخلية الحقيقية النواة. إذن الاختلاف الأساس والواضح بينهما هي وجود أو انعدام الغلاف النووي الذي يحيط بالنواة. أيضا توجد اختلافات أخرى، لكن سبب التسمية الرئيسي هو وجود أو انعدام الغلاف النووي. So, bacteria cells lack nuclear membranes, while eukaryotic cells like plant cells, animal cells, fungi, protists, these cells have nucleus that is a true nucleus because it is surrounded, because the nucleus is surrounded by a nuclear membrane. So, the cell has been the center of interest for a long time. إذن نجد أن هذه النواة هذه الخلية كانت محور اهتمام كبير من قبل العديد من العلماء لمدة طويلة. So it has been the center of interest for a long time. So the scientists have major role in development of cytology. Scientists have been interested in the cell during the past years, and they have the major role in development of cytology. So after knowing the cell and its parts, its components, and its function and its structure, so there was a new scientific science about the cell. So a study about the cell that is known as cytology. So the scientists have a major role in the development of the cytology. إذن لدينا العلماء اهتموا كثيرا بموضوع الخلية وكان لهم دور كبير في نشوء وتطور علم يسمى علم الخلية. سايتو يعني خلوي، لوجي من علوم أو علم، إذن علم الخلية. Development in cell study can be briefed as follows. إذن إحنا قبل ما نتقدم ونشوف العلماء بيش تميزوا نرجع نأكد أن هناك بعض المميزات المهمة اللي هي ما بين الخلية الحقيقية والخلية البدائية النواة. الخلية البدائية النواة كما قلنا ليست محاطة بغشاء نووي، الحقيقية محاطة بغشاء نووي، لكن أيضا هناك فروقات أخرى إضافية. إحدى هذه الفروقات أن الخلية الحقيقية النواة تكون كبيرة الحجم بينما البدائية النواة تكون صغيرة الحجم. So eukaryotic cells are larger than prokaryotic cells. So prokaryotic cells are smaller than eukaryotic cells. إضافة إلى ذلك، عندما سندرس مكونات الخلوية فيما بعد ضمن هذا الفصل، سنجد أن هناك محتويات أخرى توجد في داخل الخلايا، وهي العضيات الخلوية. بعض العضيات الخلوية تكون محاطة بأغشية، فإذن هي عضيات خلوية غشائية. هذه العضيات الخلوية الغشائية توجد في داخل الخلايا حقيقية النواة، ولا نجدها في داخل الخلايا بدائية النواة. So, as we said, the eukaryotic cells have nucleus that is surrounded by a nuclear membrane, while the bacterial cell, which represents the prokaryotic cells, has a nucleoid because the nuclear matter is not surrounded by a nuclear membrane. As well, the eukaryotic cells are larger than the bacterial cells. Bacterial cells are smaller than the eukaryotic cells. In addition, there are some parts that are found inside the cytoplasm which have the name of organelles. The organelles that are membranous, like Golgi apparatus or Golgi bodies, and the mitochondria. These membranous organelles are found inside the eukaryotic cells, but your prokaryotic cells lack these types of organelles. These are the general differences between prokaryotic and eukaryotic cells. Now we come to the scientists that aided in the development of cytology, and these scientists are as following. The first scientist that we are going to talk about is Anthony van Leeuwenhoek. The first scientist is a German scientist. This German scientist is Anthony van Leeuwenhoek. Anthony van Leeuwenhoek, as we said, he is a German scientist between the years 1632 to 1723. Anthony van Leeuwenhoek, he invented the microscope. And he was the first to see the cell. إذن العالم الألماني فان ليفنهوك أولا اخترع المجهر، ثانيا هو كان أول من رأى الخلية. إذن هو صنع مجهرا بسيط simple microscope, and he was the first to see the cell. إذن باستخدام المجهر الذي صنعه فان ليفنهوك تمكن من رؤية الخلية للمرة الأولى. إذن هذه إنجازات العالم الألماني فان ليفنهوك. ثانيا, the second scientist is an English scientist. The English scientist Robert Hooke. Robert Hooke, between the years 1635 to the year 1703. What was the observations of Robert Hooke? He had the same observations of Van Leeuwenhoek. Because he used Leeuwenhoek's microscope, so he had the same observations of Van Leeuwenhoek. And second, he was the first. This is very important. He was the first to use the word cell. What did he examine? Robert Hooke examined an oak tree cortex. This tree we usually define the parts of a tree here إذن نجد هنا أنه جاء إلى شجرة البلوط oak tree. Really, it's not the cortex here, it's the bark. إذن هو عاين وفحص القلف. هذه الطبقات الخارجية من الشجرة المعمرة يسمى القلف. إذن أخذ القلف. He took the bark, examined the bark. The bark has, when we want to examine it like this, we will find there are 3 types of tissues inside it, from the outside, the middle, and the inside. The outside part is known as cork, الفيلين. This cork, which is part of the bark of the oak tree, it is a plant tissue. He examined the cork and he drawed it. When he drawed it like that, that means, what did he see? He saw chambers. إذا ماذا وجد في هذه المنطقة الفلين؟ وجد غرف مؤلفة من غرف وقام برسمها بهذا الشكل. وهذه الغرف كانت مملوءة بالهواء. إذا ردهات هوائية. إذا وجد ردهات هوائية ضمن نسيج الفلين. This is a plant tissue. He saw these chambers. They are aerobic chambers. They are similar to the beehive. What do they contain inside them? They contain, what do they contain inside them? They contain air. So they were aerobic chambers. إذا تحتوي داخلها على الهواء وشبهها بخلية نحل العسل وقام برسمها بهذا الشكل. So he named these units or chambers, named them each of them as a cell. So he gave the name cell to the chambers that he had seen in the cork of the oak tree. إذن أعطى تسمية الخلية للغرف الهوائية الموجودة ضمن طبقة الفلين في قلف شجرة البلوط. We know that each of these units, as we said, they are cells. They are real cells. They are plant cells. إذن هذه الخلايا هي خلايا حقيقية، خلايا نباتية، لكن شاهد وجود الهواء فقط بداخلها، بمعنى أنها كانت خالية من النواة وخالية من السيتوبلازم. تحلل النواة والسيتوبلازم وجميع المكونات وبقى ماذا؟ بقى منها فقط الجدران الخارجية. في النباتات نسميها cell wall. إذن هو باعتبار أنه شاهد فقط السيل وول للخلايا النباتية. بمعنى that means when he saw these cells had no nucleus, no cytoplasm, because they have been the divorce has happened to them. That means they have been dissolved, and only the cell wall has remained. That means I can say that Robert Hooke, he was the first to see the cell wall. An information for you students, but you don't need to write it in your subject. So Robert Hooke has the same observations of Leeuwenhoek, and he was the first to use the word cell. He used the word cell on what? After he examined the cork, that means the cork, which is a plant tissue, which is part of the bark of the oak tree. The next scientist is the Scottish scientist, which is Robert Brown. Robert Hooke, Robert Brown. Robert. The Scottish scientist Robert Brown, what did he do? He discovered. What did he discover? He discovered the nucleus. إذن اكتشف النواة. العالم الاسكتلندي روبرت براون اكتشف النواة. إذا اكتشف نواة الخلية. So he discovered the nucleus of the cell. He also examined cells, and he saw the nucleus of the cell. When was that? That was in 1831. It is an important year to memorize. So he discovered the nucleus of the cell, and second, he described the nucleus of the cell. So he discovered it, but also he described it in 1831. Two important informations. After the Scottish scientist Robert Brown, we have the German scientist Matthias Schleiden. Matthias Schleiden is a German scientist. The year is very important as well, 1838. 1838, Matthias Schleiden. What did he say? He concluded. What did he conclude? He concluded that all plants consist of cells. Afterwards, another German scientist, that is Theodor Schwann, what did he do? He concluded that all animals consist of cells. That was in 1839. These 2 years are very important. So, sometimes students ask, we had Matthias Schleiden and Theodor Schwann. The both of them are German scientists. Matthias in 1838 and here in 1839. Schleiden worked on plants and Theodor worked on animals. Students ask, how can we differ between the two when we want to memorize them in the exam? You know, for Schwann, as in previous studies for you students, you had the nerve cell, you know, sorry. This is a nerve cell. It has dendrites, cell body, and axon. Sometimes we see that the axon has this part on it. Each one of those is a cell. Each one of those is a cell that surrounds the axon. This cell is known as Schwann cell because the scientist who discovered it was known Schwann, Theodor Schwann. So he named them Schwann cell. So this cell is an animal cell. So you can remember that Schwann studied on animals, while Schleiden on plants. Another thing to remember, sometimes they say, no, it's okay for these names, but it is hard for us to remember between Matthias and Theodor. Okay, I say here for Theodor, there's a cartoon that squirrels inside it. The squirrels are known as Alvin, Simon, and Theodor. So one of the squirrels is known as Theodor, has the name Theodor. Those three squirrels. So these squirrels are animals. So when want to differ between the two, both of them, you have Matthias and Theodore. And you can remember that Theodore was a squirrel in the cartoon. So when you see this name, you remember that Theodore has studied animals, not plants. That's an easy thing to remember this information. So it is very important for us to know the last two scientists in this book that has been mentioned. They are the two German scientists, Matthias Schleiden and Theodor Schwann. Afterwards, we are going to have the cell theory that these two German scientists have made, and we also have to study it. So إذا أيضا هنا عندنا كما ذكرنا مسبقا إنه بعض الطلبة يصعب عليهم تذكر شلايدن وشوان، من هو اشتغل على الأنسجة النباتية أو على النباتات ومن هو اشتغل على الحيوانات. فنتذكر بأن الخلية العصبية لها جسم الخلية والتشجرات والمحور، وأحيانا المحور يكون مغلف بهذه الأغلفة، هذه الأجزاء الواضحة. كل غلاف من هذا الأغلفة هو بالأصل هو عبارة عن خلية يلتف حول المحور، وهي الخلية المسماة بخلية شوان التي تم اكتشافها من قبل العالم ثيودور شوان. إذن خلية شوان الملتفة حول الخلية العصبية هي أيضا خلية حيوانية. إذن نتذكر بأن عندما نجد الاسم شوان نتذكر بأنه الشخص الذي درس الخلية الحيوانية أو الحيوانات. ومن ناحية أخرى أحيانا يقولون لا، إحنا المشكلة في تذكر ماثايس أو ثيودور. نقول أيضا بالنسبة للثيودور حتى تذكرها يوجد كرتون اللي هو لثلاثة سناجب، واحد اسمه ألفين والآخر اسمه سايمون والثالث اسمه ثيودور. فثيودور هو سكويرل، إذن هو حيوان. إذن أتذكر بأنه من يذكر اسم ثيودور أتذكر بأنه درس الحيوانات. نعم. إذن نجد أن هنا أن الدراسات المختلفة هنا توصلت من خلال studies and researches into cytology have increased. إذن نجد أن الدراسة هنا توسعت، والأبحاث أيضا في مجال علم الخلية. So studies and researches into cytology have increased. When did that occur? When did they increase? Particularly after advances in light microscope and invention of the electron microscope. So when I ask you, when did studies and researches into cytology increase? If it was the question as fill in the blanks with the missing words, so you will answer after advances in, first in light microscope and the second in the invention of electron microscope. إذن متى توسعت الدراسات والأبحاث في مجال علم الخلية؟ حدثت بعد تطور المجهر الضوئي واختراع المجهر الإلكتروني. إذن عندنا متى حصل increase ب studies and researches. We say here after advances in light microscope، وأيضا عندنا after invention of electron microscope. The study has become a major branch called cytology. إذن أصبح هناك فرعا رئيسيا من فروع علوم الحياة يعرف بعلم الخلية. So when did cytology become a major branch? when did cytology become a major branch? That is, cytology have increased and developed, particularly after advances in light microscope and invention of electron microscope. So cyto, that means cell, logy, which means the science. So we say cytology. إذن علم الخلية جاء من كلمتين، cyto هي خلية ولوجي هو العلم. نأتي بعد ذلك إلى موضوع آخر وهو the cell theory. The cell theory. So the second important thing we are going to talk about is the cell theory. إذن عندنا نظرية الخلوية أو نظرية الخلية. فلدينا عندنا العلماء كلاهما العالم الألماني ماثايس شلايدن والعالم الألماني ثيودور شوان هم من أسس هذه النظرية العلمية. So this scientific universal theory by the two German scientists Matthias Schleiden and Theodor Schwann has mainly states three tenets. That means it has three parts. There are three parts of this cell theory, and the two German scientists led to the foundation of the cell theory after they studied on plants and animals. So they wrote this theory, the scientific theory. So this theory, what does it state? First, it states that all organisms are made of cells. Yes, if we come to plants, to animals, to fungi, to protists, bacteria as well, we can see that these organisms may be made of only one cell, unicellular, or sometimes they are made from more than one cell, so they are multicellular. So that's why they say that all organisms are made of cell or cells. The second part of the cell theory is that cells are the basic structural and functional units of all organisms. إذن عندنا هنا أن الخلايا هي الوحدات الأساسية التركيبية والوظيفية للكائنات الحية. بمعنى إذا كان عندي مثلا هنا مجموعة من الخلايا أو كانت خلية واحدة، هذا الكائن الحي المتكامل، عندما عندي مثلا كائن حي متكامل، هذا الكائن الحي المتكامل كل مبني من خلية أو من خلايا مع بعض. فلولا وجود هذه الخلايا لما كان أصبح هذا الكائن المتكامل. إذن الخلايا مع بعض تجمعت لتكون هذا الكائن المتكامل. إذن هي الوحدة بناء هذا الكائن، the structural unit، وحدة بناء. ونفس الوقت أنه كل خلية هنا تقوم بوظيفتها، فعندما تقوم كل خلية بوظيفتها بالنتيجة فإن هذا الكائن أجمع يقوم بالوظائف الخاصة به. إذن لولا كل خلية تقوم بوظيفتها لما كان هذا الكائن قادر على القيام بالوظائف المتكاملة له. إذن هي وحدة بناء الكائن ووحدة وظيفة كل جزء من أجزاء هذا الكائن الحي. So, as we said, an organism is made of one cell, or he is multicellular. That means some of the organisms are made only of one cell, and others from more than one cell. Also, the second part talks about the structural unit of this organism. What is the structural unit of it? It is the cell. Each cell of this organism form this the structural parts of this organism. That it means this organism is made of these cells. Without these cells, there will not we will not see this organism, or this organism does not exist. So this organism is made of all these cells together that have gathered, the cells gathered together to form this organism. Each of these cells has a specific work to do. So when this cell does its function, and all these cells as well do their function, so the organism can live properly and do all its functions properly, because it depended on each of these cells. So the cell is the structural unit and the functional unit of all the organisms. The third part talks about this cell. How does it originate? So if I had this cell, how does it result into these many cells? That is, this cell divides. When a cell divides more and more, it will form these multiple cells. So each cell has originated from a pre-existing cell, from a previous cell. So this cell has came from this cell. These two cells have came from this cell by means of cell division. And how did that happen? By cell division. So the origin of a cell is from a previous cell by cell division. إذا منشأ هذه الخلايا من أنتجت من الانقسام الخلوي لهذه الخلية السابقة. إذا هي لم تأتي من العدم، وإنما وجود هذه الخلايا جاء من الانقسام الخلوي للخلية سابقة موجودة. إذا عندنا هنا أيضا ممكن يكون عندنا سؤال عن منشأ الخلية. إذا نقول what is the origin? The question, what is the origin? What is the origin of a cell? The answer will be one cell comes from another. One cell comes from another cell. How? By means of cell division. So a cell comes from another pre-existing cell by cell division. So this is the cell theory. It is also important. And if we ask you at the exam, what is the cell theory? The answer will be the cell theory is mainly based on the work of both Matthias Schleiden and Theodor Schwann, which can be briefed as follows. And you mention these three important parts of the cell theory. And when I ask you about the origin of a cell, you say it comes from another cell by means of cell division. Who is responsible for this theory? The two German scientists, Matthias Schleiden and Theodor Schwann. Now we come to another important title. It is the cell size. Cell size. Do cells have the same size? No. There is difference between cells and size. So cells vary in size. For example, diameter of frog's egg is 1 millimeter. That means we can see these cells by our naked eyes. So, maybe not all of you have seen frog's egg, but some of you maybe have seen fish egg. When we buy a fish from the market, and we open this fish at home, we cut open this fish at home, sometimes we can find these parts inside it. These are eggs inside the fish. If you remember, when you saw these eggs like that, it is small and spherical and small, and when I want to count them, they are hundreds, maybe thousands. But what do I recognize? That each one of them is spherical. It's an egg. It's an eukaryotic egg. It's a true egg. So it has a diameter that is probably nearly to the frog's egg. So I mentioned the fish egg because maybe a lot of you have seen it, but frog's egg, not all of you have seen it. So when I recognize it, this egg of the fish, maybe I can say that it has a diameter of 1 millimeter. These eggs, I can see them by my naked eye without the use of microscope. So that means if I'm going to ask you, how does cell size vary? If my question was, how does cell size vary? What would be your answer? The first answer would be diameter of frog's egg is 1 millimeter. So that, what does it mean? It means it can be seen by naked eyes. So some eggs, that means some cells that can be seen by naked eyes without the use of microscope. إذن هنا عندما نأتي إلى كما ذكرت مسبقا أنه السمك عندما نقوم بشرائه من الأسواق، وذكرت مثال السمك لأنه أغلبكم ممكن تكونوا شاهدتوا السمكة، لكن القليل من شاهد بيض الضفدع. فنجد أنه السمكة عندما نقوم بشرائها ثم نفتح هذه السمكة نجد أحيانا بداخلها بيض السمك. بيض السمك أحيانا يسمى بالثرب بالعامية. هذا البيض عندما أركز فيه أجد أن هناك خلايا دائرية كروية الشكل صغيرة الحجم، لكن أراها بالعين المجردة. صحيح لا أرى مكونات الداخلية لكن أراها. إذن هذه الخلايا ترى بالعين المجردة، والتي هي قريبة من حجم بيضة الضفدع، وقطرها يبلغ 1 مليمتر. إذن عندما يسأل عن هل تختلف أحجام الخلايا، نعم تختلف. في بعض الخلايا مثل الخلية اللي هي بيضة الضفدع هي خلية قطرها يبلغ 1 مليمتر، وممكن أن أراها بالعين المجردة دون استخدام المجهر. The second answer that is added to this question, most of cells. So here, most cells are smaller than 1 millimeter. An example, we have a human egg. It has a diameter about 100 micrometer. or less than 100 micrometer. As a note, 100 micrometer, that means it is 1 to 1,000 millimeter. So you can compare between the two, and you see that the frog's egg is larger and bigger than a human's egg. So the human egg has a diameter of 100 micrometer and sometimes less than that. So the other answer here, we're going to give it to this question, is most cells are smaller than 1 millimeter. An example, we have a human egg is about 100 micrometer or less. So we gave here two examples about how does cell size vary. Also, it is an important question. When we come to cells, for example, animal cells, we find there are nerve cells, muscular cells, bone cells, cartilage, and so. So these cells differ from each other in shape, in function. So cells have specific specialities to increase its efficiency for various functions. إذا تظهر بعض الخلايا تخصصات معينة. ليش؟ لماذا تظهر تخصصات؟ يعني معناتها بيها بالأمور مميزة بداخلها بما فيها الشكل. يعني شكل الخلية العصبية يختلف عن شكل العضلية، ويختلف عن العظمية وعن الغضروفية وعن باقي أنواع الخلايا. أيضا هذا الشكل له دور العلاقة بالوظيفة اللي تأديها هذه الخلية، وأيضا من أجل زيادة كفاءة الخلية في أداء عملها. إذا لزيادة كفاءتها في إنجاز الوظائف المختلفة، فنجد أن هذه الخلايا تظهر تخصصات معينة. إذا عندما يكون السؤال explain the cells, why do they have specific specialities? The answer would be is to increase its efficiency for various functions. Now we come to this part. We need microscope. We need microscope. Why? To see cell and what? And also to see microorganisms. Cells can be seen by using light microscope, but inner components of the cell like organelles, viruses, and organic particles can only seen by the electron microscope. So sometimes I can see an egg, I can see a cell, but I cannot see what's inside it. So I need an electron microscope to see the components clearly inside these cells. So the question will be, what are the importance of microscopes? Or what is the importance of microscopes? So the answer will be, we need microscopes to see cell and microorganisms. So first, cells can be seen by light microscope. And second, I will add it to the answer, that is inner components of cell like organelles, viruses, and organic particles can only seen by electron microscope. This is the answer for the question. Now we come to an important formations we're going to take about prokaryotic cell. After we said there are two types of cells, prokaryotics and eukaryotic cells, first we're going to take the prokaryotic cell. What is a prokaryotic cell? It is an undeveloped cell. It is more primitive in shape and structure and characterized by the followings. إذن الآن عندما نأتي إلى الخلايا بدائية النواة فنبدأ بذكر أهم مميزات الخلايا بدائية النواة. فإن هنا نذكر تعليل مهم إنه الخلايا البدائية النواة تعتبر undeveloped يعني بدائية أو خلايا أقل تطورا من الخلايا الأخرى. لماذا؟ إذن لماذا تعتبر الخلايا البروكاريوتيك undeveloped cell؟ الجواب لأنها خلايا أكثرها تكون بدائية. بدائية من حيث ماذا؟ من حيث الشكل، من حيث التركيب، وتتميز بالتالي. إذن لما يكون السؤال هنا What are the characteristics of prokaryotic cells؟ راح نبدي ندرج شنو هي المميزات الموجودة ضمن الخلية بدائية النواة. لكن عندما يسألني Why are prokaryotic cells اعتبرت يعني why are prokaryotic cells considered as undeveloped cells? So the answer would be because it is more primitive in shape and structure. Why is a prokaryotic considered as an undeveloped cell? الجواب because it is more primitive in what? In shape and in structure. Now we are going to mention the characteristics of prokaryotic cells. Prokaryotic cells has genetic material without a membrane, and it is called as nuclear zone or nucleoid. We mentioned that previously, that prokaryotic cells have nuclear matter, but it is not surrounded by a nuclear membrane. So what do we call it here, this nuclear matter? We can name it as nuclear zone or nucleoid. إذن الخلية بدائية النواة هنا تحتوي على مادة وراثية genetic material، لكن هذه المادة النووية أو المادة الوراثية ليست محاطة بغشاء نووي، ليست محاطة بغشاء، لذلك تسمى بالمنطقة النووية هنا أو بمنطقة النواة أو المنطقة النووية، لكن هي ليست نواة حقيقية، وإنما ذكرنا مسبقا هي نواة بدائية. The second characteristic here is prokaryotic cell has cytoplasm, and this cytoplasm lacks or hasn't any membranous organelles. These organelles that are membranous, like Golgi bodies, or sometimes we call them Golgi apparatus. And the second organelle mentioned here is the mitochondria. So prokaryotic cells lack Golgi bodies and mitochondria. while it has ribosomes. Note, ribosomes are non-membranous organelles. This ribosome in the form of numerous small grains. They are responsible for building proteins. إذن هنا نجد أن الخلية البكتيرية أو الخلية البدائية النواة تحتوي على السيتوبلازم، لكن سيتوبلازمها لا يحتوي على عضيات غشائية. من هي هذه العضيات الغشائية؟ سنأخذها فيما بعد إن شاء الله. مثال عليها جهاز كولجي أو نسميها أجسام كولجي، وأيضا الميتوكوندريا التي هي بيوت الطاقة. هذه عضيات غشائية توجد في سيتوبلازم الخلايا حقيقية النواة، لا نجدها في الخلايا بدائية النواة. لكن الخلايا البدائية تحتوي على بعض العضيات في السيتوبلازم التي نجدها في البدائية ونجدها في الحقيقية. من هي هذه العضيات؟ هي الرايبوسومات. هذه العضيات ليست لها أغشية، ليست محاطة بأغشية، فهي لا غشائية. وهذه الرايبوسومات توجد بهيئة طبعا كثيرة العدد وبهيئة حبيبات small grains. ما وظيفتها؟ وظيفتها بناء البروتينات في داخل الخلية. إذن عندما يسألني من هو المسؤول عن بناء البروتينات في داخل الخلية البكتيرية، هنا المسؤول عنها هو الرايبوسومات. عندما يسأل ما نوع العضيات الموجودة في داخل سيتوبلازم الخلية البدائية، نقول الرايبوسومات. ما هي العضيات الغشائية المفقودة في داخل الخلية البدائية؟ جهاز كولجي والميتوكوندريا. And next we come to the C. Prokaryotic cell is represented by blue-green algae, bacteria, and mycoplasma. These three examples belong to the kingdom Monera. إذن هنا نعطي أمثلة على الخلايا البدائية النواة المتمثلة لدينا هنا الطحالب الخضراء المزرقة. الطحالب الخضراء المزرقة هي أيضا بكتيريا، احنا نسميها أيضا بالسيانوبكتيريا. هذه البلو جرين ألجي هي سيانو بكتيريا، هي بكتيريا لكن تحتوي على كلوروفيل فتقوم بعملية البناء الضوئي. المثال الآخر أن البكتيريا بأنواعها المختلفة، وإضافة إلى أن المايكوبلازما أيضا هي بدائية النواة، وجميعها تعود إلى مملكة المونيرا. إذن هذا أيضا مهم. إذا يسألني عن العالم الذي يعود إليه البلو جرين ألجي، أو يسألني عن عالم البكتيريا إلى أي عالم تعود إلى عالم المونيرا. أو إذا يسألني عن المايكوبلازما، إذا كانت الفراغات أو مثلا بالامتحان، أو ممكن يكون example, give me an example of for monera. فكل هذول الأنواع مطيني هي تعود إلى عالم البدائيات اللي هو المونيرا. So here it mentions bacteria differ from blue-green algae because they have no chlorophyll. He mentions here that bacteria does not have chlorophyll pigments, while blue-green algae has chlorophyll pigments. And also here, cell wall of bacteria is surrounded by a capsule. We're going to take it afterwards. While blue-green algae is surrounded by a gelatin membrane. So there is a difference between the blue-green algae and the bacteria. The first difference is the blue-green algae has chlorophyll pigments, while bacteria does not. Also, the blue-green algae is surrounded by a gelatin membrane, while the bacteria is surrounded by a capsule. As a note, not all the bacteria are surrounded by capsule, but some of them are surrounded by capsules. Some other types are not surrounded by capsules. So when the question that I ask you is, what are the characteristics of a prokaryotic cell? What is the answer that I need? The first answer is A here, the second is B, and the third is C. Also here, we have other characteristics that are important inside this bacterial cell or that inside the prokaryotic cells. Each bacterial cell represents a prokaryotic organism. إذن تمثل كل خلية بكتيرية كائنا بدائي النواة. What is this cell surrounded by? It is surrounded by a solid wall. Consist of chemical compounds. What are these compounds? They are protein, lipids, and polysaccharides. Behind this wall, there is the plasma membrane, which is semi-fluid membrane. What does it do here? It surrounds the cytoplasm. And the cytoplasm contains the nucleoid. As we know, there are no nuclei or nuclear envelopes. Unlike eukaryotic cells, cytoplasm also contains ribosomes. Some types of moving bacteria have flagella, pili, or both. So here, when we come to the bacteria, it is important to know these. As we said, prokaryotic cells like bacteria, what do they have? They have the cytoplasm. The cytoplasm is surrounded by plasma membrane. Sometimes the plasma membrane here is surrounded by cell wall, and not all bacteria is surrounded by a capsule, but some of them are surrounded by a capsule. And here we recognize that there is a genetic material here, which we named it as nucleoid. That lacks the nuclear membrane. Another information that I didn't mention before, that when we compared it with eukaryotic cell, the eukaryotic cell has plasma membrane that is not surrounded by a cell wall. But we have examples that of eukaryotic cells that are surrounded by cell wall, that are the plant cell. Also, fungi is surrounded by cell wall, but it is not surrounded by a capsule. It has cytoplasm inside it. It has a nucleus. So this nucleus has genetic material, but it is surrounded by a nuclear membrane. Also, there is a part here which is very important, which is known as nucleolus. Nucleolus النوية. The nucleoid here in bacteria does not have nucleolus. So the nucleus in prokaryotic cells lack the nuclear membrane, also lacks nucleolus. إذن هنا عندما جئنا إلى الخلية البكتيرية هذه المادة النووية هنا ليست محاطة بغلاف نووي، وإنما توجد في داخل السيتوبلازم. هذا السيتوبلازم محاط بالغشاء البلازمي، بلازما ممبرين. الغشاء البلازمي محاط بسيل وول، جدار خلوي. أحيانا البكتيريا تكون محاطة بكبسول، محفظة. ليست كل البكتيريا محاطة بمحفظة. عندما قارناها مع الخلية الحقيقية، قلنا الحقيقية لها غشاء بلازمي يحيط بالسيتوبلازم، وهي ليست محاطة بجدار خلوي، لكن الخلية النباتية والخلية الفطرية محاطة بجدار خلوي. عندما أتينا إلى النواة، نواة حقيقية لأنها محاطة بغشاء نووي، وأيضا يوجد شيء آخر وهي النوية. النوية موجودة في نواة الخلايا الحقيقية النواة ومفقودة في الخلايا بدائية النواة. إذن هنا تفتقد إلى النوية وتفتقد إلى الغشاء النووي. مكونات الجدار الخلوي أيضا تختلف ما بين الحقيقية وبدائية النواة. عندما نأخذ الجدار الخلوي في الخلايا النباتية أغلبه يكون من أسيلوز، يعني السكريات المعقدة، وقد يضاف إليه الشمع كايتين. لكن عندنا بالفطريات الجدار الخلوي تركيبة نوعا ما باختلاف عن مكونات الجدار الخلوي في النباتات. أما هنا عندما نأتي إلى الجدار الخلوي في البكتيريا نجد أنه يتألف من 3 مكونات رئيسية، هي البروتينات والدهون ومتعددة السكريات. So when we mention the cell wall here, cell wall in plants is composed of polysaccharides that is in the formation of that forms cellulose, while in fungi has different components from plants, the cell wall. Here in bacteria, cell wall is composed from 3 important components, which are proteins, lipids, and polysaccharides. If this bacteria has the ability to move, so it needs something to move with. These things, maybe they are short and many, they surround the capsule, which are known as pili, or sometimes we name them as cilia, or they have another name which is fimbria. So it has 3 names: pili, cilia, or fimbria. They are short extensions, short extensions, but they are many in number. Or it has a long extension, which is known as flagella. This long extension may be 1, maybe 2, maybe 3, maybe from both sides, but they always are small in number, easily to count. But cilia can reach hundreds, thousands of cilia or pili or fimbria, as we said. But both of them, cilia or flagella, are used for moving. So it is very important to ask what are the chemical components, or what is the chemical composition for the cell wall of bacteria, or bacterial cell wall. So a question like that will be, what is the chemical composition of bacterial cell wall? The chemical composition of bacterial cell wall. The answer would be, it is consisted of what? Of these chemical compounds that are protein, lipids, and polysaccharides. This is very important. So if he asks me about or to mention the characteristics of prokaryotics, we are going to mention all of these informations. What are these informations that we are responsible of to mention them in the examination? The first important thing we say it is a primitive, and it is more primitive and has a genetic material without a membrane, and it is called nucleus zone or nucleoid. Prokaryotic cell cytoplasm has no membranous organelles like Golgi bodies and mitochondria. Third, it has ribosome in the form of numerous small grains which build proteins. Fourth, prokaryotic cell is represented by blue-green algae, bacteria, and mycoplasma, which all belongs to kingdom Monera. The fifth, plasma membrane is semi-fluid membrane, surrounds the cytoplasm, which contains the nucleoid. Sixth, there are no nuclei or nuclear envelope. Seventh, some types of moving bacteria have flagella, and some of them have pili or both. Eighth, it is surrounded by a solid wall, and this wall is composed of protein, lipids, and polysaccharides. These important informations here: the general appearance of bacterial cell. What are the main structures that we can find in a bacterial cell? First, it has a cell covering. Second, it has cytoplasm. 3, it has these suffixes. As mentioned here, cell covering may be cell wall, plasma membrane. Cytoplasm, it has nucleoid and ribosomes as organelles. The suffixes, if it has the ability for moving, maybe it has flagella or it has fimbriae. Or sometimes it has a sex pili. A sex pili, usually we are going to take it at this information at chapter 3 that talks about reproduction. So in reproduction in bacteria, we will find that the male bacterial cell has a sex pili that aids in sexual reproduction. Not all bacteria has it, but during sexual reproduction, only the male bacteria has sex pili. This drawing here of the structure of bacteria is very important. And how are we going to draw it? First, this bacterial cell, we are going to open it to see what's inside it. This is the cytoplasm. Inside the cytoplasm, as we mentioned before, it has a zone here that we can name it as nucleoid. That is not surrounded by a nuclear membrane. Here, this cytoplasm is surrounded by a cover. This cover, here I'm going to make it more clear. This cover, this is part of the cover here, is the plasma membrane. The second cover here that surrounds the plasma membrane is the cell wall. This is the cell wall. Which consist of chemical compounds. They are proteins, lipids, and polysaccharide. The cell wall, this cell wall is surrounded by a capsule. This capsule surrounds the cell wall. This capsule sometimes has fimbriae, cilia, a lot of them, many extensions, many short extensions, which are known as fimbriae. Sometimes this bacterial cell has a single flagella. Sometimes it has more than one. Sometimes it has cilia and flagella, the both of them together. Also, we said it has organelles like ribosomes that are responsible for protein synthesis. And as a note that is important, the pili and the flagella, they extend to reach the cytoplasm. That means they extend from the cytoplasm and through plasma membrane, through the cell wall and the capsule. Also the sex pili. This sex pili, sex pilus, extends from the cytoplasm and through plasma membrane, cell wall and capsule. It helps the male bacteria cell in sexual reproduction. This is very important and easy to draw. إذن الخلية البكتيرية هنا تتألف من السيتوبلازم، والسيتوبلازم بداخله المادة الوراثية genetic material سميناها نيوكليويد، ويوجد أيضا في السيتوبلازم العضيات غير الغشائية وهي الرايبوسومات لبناء البروتينات. السيتوبلازم هنا محاط بالغشاء البلازمي. الغشاء البلازمي محاط بالجدار الخلوي، وذكرنا مكونات الجدار الخلوي، والجدار الخلوي محاط بالكبسولة المحفظة للحفاظ على جميع مكونات الخلية البكتيرية. يمتد أحيانا من البكتيريا إذا كانت لها قابلية على الحركة، يمتد منها الأهداب التي تمتد من السيتوبلازم ونحو الخارج، فتخترق الغشاء البلازمي والجدار الخلوي والكبسولة. أو الصوت الفلاجيلا أيضا ممكن يمتد من السيتوبلازم ويخترقها جميعها فتستخدمها للحركة، فممكن يكون لها فقط أهداب أو ممكن يكون لها فقط صوت أو ممكن تحتوي على الأهداب والأصوات، وأحيانا لا تحتوي لا أهداب ولا حتى أصوات، فهي تكون خفيفة الوزن وينقلها الهواء وينقلها الماء. وأيضا قد تكون لها من ضمن الملحقات السفكسز هنا الملحقات إنه تكون عندها هذا اللي احنا نسميه بالهلب الجنسي، وهو توجد عند الخلية الذكرية البكتيرية الذكرية، نذكرها لاحقا إن شاء الله في موضوع التكاثر بما فيه التكاثر في فصل التكاثر الفصل الثالث ضمن موضوع التكاثر في البكتيريا، بالأخص في التكاثر الجنسي. سنلاحظ الخلية الذكرية لها سكس بيلاي يساعدها في عملية نقل المعلومات الوراثية إلى الخلية الأنثوية. إذن هذه الرسم جدا مهم. As we mentioned, some bacteria does not have cilia or flagella, but they are so light so that are easily to be transported by air or by water. The last information we are going to have today is we are going to talk about the eukaryotic cell. As we mentioned before, it is a cell that has a true nucleus, and this cell is found in which type of organisms? After the kingdom Monera, we're going to take the protists and fungus and plants, finally animals. So eukaryotic cells found in protists, fungi, plants, and animals. These organisms have true nucleus. That means the nucleus is surrounded by a nuclear membrane. Eukaryotic cells differ in shape. That means they do not have the same shape. So when I ask you, does all cells or all organisms have the same types, the same cell shapes? No. The answer is no, they do not. Even when we come here to the animals, like in human being, does the nerve cell, muscular cell, cartilage cell, do they have the same shape? No, they do not. Because the shape is responsible for the type of function. The cell needs a specific function, a specific shape to do its specific function. So when I ask you what types of shapes that we have in these different organisms, in these different kingdoms, we have about, here it mentions about 10 types of shapes. We have the spherical, pyramidal, the tubular, cubic, oval, flat, astral, fusiform, and also some have varying shape that changes its shape. So here we mentioned the different types of different shapes for different cells. We have the spherical، عندنا هنا أنواع مختلفة من أشكال الخلوية. عندنا spherical التي هي الكروية، pyramidal اللي هي الهرمية، tubular اللي هي عندنا الأنبوبية، وكيوبيك المكعبة، oval البيضوية، وفلات التي تكون مسطحة، astral اللي هي النجمية، fusiform اللي هي المغزلية، وvarying in shape التي هي تتخذ أشكالا مختلفة. من يتخذ أشكالا مختلفة كما لدينا في عندنا الوايت بي سي، كرات الدم البيضاء، وأيضا عندنا ناخذ الكائن أميبا اللي هي نوع من أنواع البروتيستس. هذه أيضا تكون لها تغير شكلها، إذا ليس لها شكل ثابت. إذا ممكن يطلب مني أشكال الخلايا فأعدد هذه الأشكال. When it asks me about the different shapes of cells, I'm going to mention all these types of cells. So this change in shape، هذا التغير بالشكل، يعني كل خلية لها شكل مختلف، إلى علاقة بماذا؟ إلى علاقة بالوظيفة، وهذه جدا مهمة. إذا الشكل المختلف له علاقة بالوظيفة المناطة بهذه الخلية. So when I ask you a question that is why do cells have different shapes? The answer would be because they have different shapes, because the shapes adapts the functions for these cells. So cells often have shapes that adapt their functions. So that's why cells have different shapes from each other. We mentioned also eukaryotic cells are small and can only be seen in microscope. We mentioned that eukaryotic cells are larger than prokaryotic cells, but also they are small in size. But they are larger. They are small, yes, but they are larger than prokaryotic cells. Cells need surface area. What is the surface area that a cell has? This is the cell. It is surrounded by what? It is surrounded by a plasma membrane. So the plasma membrane is the surface area. Why is it important for a cell? Why does a cell need a surface area? It helps in the exchanging of materials. It needs to exchange materials between the environment inside it and the environment outside the cell. So for the exchange of materials with the surrounding environment, it does it properly. Why does it do it? Because it has a surface area that is represented by the plasma membrane. It is needed for every cell. In the next lecture, we are going to talk about eukaryotic cells and what they consist of components. We are going to take them briefly. Until now, we reach to the end of the lecture for today. So goodbye.